This is a relatively, I think, simplistic question, but it helps sometimes just to summarise in one's own head when there's been quite a lot of information. Is the, excuse the pun, the gist of what you're saying, that you're trying to work to get away from a one kind of tablet fits all?
Hopefully, yes.
That's the end goal.
Yeah, that's the end goal.
Yeah.
I think that would be, perfect world where we can just look at the tumour profile, look at the blood tests and say, well, this is the ideal treatment for you as an individual person. I think, we're a little bit away from that right now, but I think if we can be a little bit more selective, then we can build on that and, hopefully one day we'll be able to.
Although when you put it in the context of 100 years ago, it probably was surgery and nothing. So we're doing pretty well.
Well, indeed, and in a way, I mean, the whole imatinib in GIST story just highlights that. Prior to the publication in Science in 1998, showing the KIT mutation in GIST, it was a really, really rare diagnosis, but with an immunohistochemical, with a protein test that we could use to, in a way, as a surrogate, and then a drug being developed for another disease that actually targeted that, not only has it led to all of these drug approvals, but it's also shown us that GIST, in fact, isn't a rare disease, and for all sarcomas, it's probably one of the more common subtypes, probably about 20% of sarcomas. So you're absolutely right. It's collaboration and working together to make things better, and that can take time. But realistically, 1998 well, it is a long time ago, and the world has changed a lot. But it's still a relatively short period of time. But
that was a massive leapfrog, and there's every hope with the work that you're doing that exponentially.
Yeah, you're right. It's sort of incremental in a way, rather than a massive leap forward. But I suppose often there are incremental steps, and sometimes there are massive leaps forward.
Yeah.
But, yeah. Don't get me wrong. I'd love to have more massive leaps forward. But, yeah.
Thank you very much.
Hi, thank you. I've got a question for Zoe.
Sorry, we haven't seen each other for ages.
And it's partly my fault because we were due to meet up, and my schedule's just been a little bit crazy. So my apologies for that as well, Zoe. I've said it publicly.
You heard it here first.
So you had a slide with the 2 patients and 7 patients.
Yeah.
And then down the right-hand side, there was the breakdown of types of cells.
Yeah.
That were kind of obviously reducing down to the killer cells right at the bottom. So the GIST cells, part of the way down there, is that what indicates the mitotic count of tumours?
No, that's literally us just saying, can we see a tumour cell in this big sort of soup of cells that we get through?
Okay.
So the way these things work is we get a piece of material and then we have to kind of disassociate it into all its single cell components and then run it through a machine, and then essentially we just count how many of those different cell types we see when we do that. And so actually what we're finding, which is quite interesting, is that rather than it being 90% tumour cells, there's lots of other different, these normal cell types, these immune cell types, these what we call stromal cell types that are supportive, and we see lots of those different cell types. And really, I guess from a kind of what does it mean perspective, it is going to allow us to understand exactly how the cancer cells are talking to and interacting with them. And what's really interesting is that a lot of what we talk about is talking about targeting tumour cells, but we now know that actually you can give drugs that will target some of these other cells that will allow things like chemotherapy to work better. So there are cells called fibroblasts that are these supportive cells, and they have a particular type when they're associated with cancer cancer, and you can actually give, believe it or not, things like Viagra to change the way that, I know, right? But you can actually change those fibroblasts to a different, so they basically change their shape, and it actually allows things like chemotherapy to work better, and we've shown that in some other solid tumours. And the more we understand about GIST, the more we can say, oh, that they're doing in a different cancer type might work for this, because we're seeing something similar. And it opens up those options that are not just, can we get a small molecule inhibitor that targets a tumour cell? Can we adapt that tumour microenvironment to let things we've already got that work better?
Yeah, thank you.
I'm told Viagra has a good way of changing shape.
Hi, thank you very much for that. I noticed just about 3 slides before the end on the table that right at the bottom, in what appeared to be the driver mutations, you'd got unknown at the bottom. Does that mean that you are trialling some of these combination therapies on the wild type GISTs that whereby the driving mutation is unknown?
So that's a really good...
And if so, what responses are you getting?
So that's a really good question. One of the, I suppose one of the challenges with doing large international trials at multiple centres is sometimes, the information's not available. In some countries, actually, particularly the USA, mutational testing isn't done very often. There are challenges with doing mutational analysis in community oncology centres, and sometimes that information is just not available. Usually, though, the entry criteria for clinical trial, and for most of the clinical trials I mentioned, had specific inclusion-exclusion criteria for particular mutational subtypes, basically. So I think that's just one of those challenges with international trials, that sometimes patients with tumours that haven't been properly characterised are included into trials. So I think things are definitely improving in that respect in terms of mutational analysis being standard, but it's still a problem in a way in certain geographic areas. Some of the trials have included KIT and PDGFR alpha wild type, but I'd sort of, in a way, question the rationale or the logic of that. But I think increasingly it's becoming more focused on specific mutational subtypes, as we discussed, so that we can sort of refine treatment in the future. But well spotted. That's it. Here's an old friend.
Oh, yes, one of the longest. The trials using different lengths of time for imatinib in adjuvant treatment. First of all, 2 questions really. The data that you've presented is for time to recurrence. Have you yet got data of time overall survival?
So for the French trial, I think we need a little bit more time for overall survival. They've stated that there's no difference at present, but I think that's in a way one of the crucial questions about that trial, and I think it will need more time, because even if people go on to develop metastatic disease, with so many treatments available to treat metastatic disease now, people are living for longer and longer, which is a good thing, but it means that trials take longer to analyse for overall survival in particular.
Because, patients have to put all the data together, the effects on their life if they're taking imatinib, and if it's not going to extend their life, they might prefer not to take it, and have a better quality of life, and rather than living a life with the drug. And if it's not going to make any difference to how long they live, why take it?
Well, exactly. So I think again, it's fairly early days with the French sarcoma group trial that they've reported no difference in quality of life. But I'd say let's see what the subsequent publications show. I think very often this comes down to an individual discussion with an individual person regarding the the pros and cons, particularly if if the risk is really, really high, or if the risk is less, then it can be a much more subtle discussion. And as you know, it's a discussion that we have every day almost in clinic, but a very, very valid point.
My other question really is, if you're taking the drug adjuvantly, do you still take the same dose? Do you take a lower dose? And how is that decided?
Yes. So again, a great question, and the standard dose, of course, is at 400 milligram. But very often we will decrease the dose depending on side effects, depending on tolerability. Some people use blood tests to measure the imatinib levels. When the cancer spread in the metastatic setting, I'd usually just go with side effects and just adjust the dose appropriately. And we've got people who've been on imatinib for 10 years plus just on 100 milligrams. So there's a great deal of variation. And in a way, it comes back to your question regarding trying to sort of personalise the treatment more. And I think we're still a long way off in terms of the that sort of very refined dosing and drug selection for an individual patient. But it's something that we should be really aiming for.
It's a long-term process, isn't it?
Indeed.
Yeah.
And I think you're absolutely right. The 400 milligram dose isn't the right dose for everybody. And as I say, we've got patients on 100 milligram for 10 years, 15 years plus.
But in an adjuvant treatment, the patient might decide, I don't like 400 milligrams. Can I try 200? But then, of course, it messes up your statistics.
Yeah, but it's a discussion that that I do have with people all the time. And in a way, right now we don't have a right or wrong answer. It's a case of weighing up with the individual person. So, yeah, good. I
turn you in a second.
Too soft. I need to shout more.
A question maybe some of the audience might be wondering is, how are things looking from a side effects point of view with the now imatinib is off patent?
Yeah, so some of the generic preparations, I must admit, I've in a way been quite disappointed that they do seem to have more side effects. We're trying to sort of work through that as well, and we have changed a number of people back to the preparation that seems to be the right fit for them. But I agree, it's been disappointing that some of the generic preparations have been quite toxic, really, and people have noticed the difference immediately. So
and how easy is it for a patient that is suffering toxic reactions to generic to change to another generic within their oncologist area?
So I suppose in a way I can only speak with regard to where I work, and it's been relatively easy, and we've been working with pharmacy to just find the right formulation for the individual patients. So the short answer is I think it should be relatively easy to do that.
So a patient's perfectly at liberty to say,
yeah, of course, yeah.
I'm not getting on with this. Can I try something else?
Definitely so.
So
There's a question at the...
Yeah, Tony behind you first.
Oh, yeah. Hi,
Sorry,
Tony.
I'm
Hi.
going to turn around. I guess my question is, it's kind of related to both of the talks. So, I mean, Zoe obviously was talking about identifying areas that can be targeted that are not necessarily the tumour cells, and therefore there might be existing off-patent drugs or treatments for other which target the same sort of microenvironment. The biggest challenge that I see with these new drugs that we're talking about trialing, and some of the ones you mentioned that are now, approved by the FDA, is that they don't get approved by NICE, normally because they're too expensive. So I guess my question is do you think that it's more beneficial that if we can find drugs that are already in existence and repurpose them, then maybe it means that there's more treatment opportunities?
Yeah, I think that's a fantastic question. I think the answer is yes. That would be fantastic. I'd say, from experience with another type of sarcoma where we have tried to repurpose an old drug, a really, really rare type of sarcoma, it's incredibly challenging to go through the regulatory process for repurposing older drugs. And there are several problems there, or several hurdles in terms of the quality of data. Coming back to Judith's point, the quality of data from retrospective studies, so going back and looking at notes and trying to sort of collate and record what the response and the side effects and the benefit from a particular drug were, compared to collecting data prospectively as in a clinical trial. And from several of these meetings with the EMA and FDA, I think everybody is very, quote-unquote, supportive of trying to do this, but trying to sort of pin down the regulatory agencies in terms of saying, well, what is the sort of benchmark in terms of quality of real-world data that you want? It's really, really difficult. And with that, sometimes I get not angry, but just it's a little bit frustrating because you can never get a straight answer regarding what is the quality that you actually want for retrospective data. And the other thing is it's quite difficult to get, if it is a drug, it's quite difficult to get a pharmaceutical company interested in a relatively new, maybe even expensive process of repurposing a drug like that. So I think it's a fantastic idea and a good thing to try and do, but there are definitely challenges in terms of getting that done. And even doing a prospective trial of an older repurposed drug would be expensive and could take a lot of time. Yeah.
I totally agree with everything Robin said. I think one of the things, maybe this is sort of a way of reassuring you, one of the things we try to do in our team is think about that sort of thing. And actually, often it's not necessarily an older drug. It might be a drug that's currently already in a phase 2 trial or something where we think we might have a bit more potential to have those conversations with the pharmaceutical company at that point of where they're trialing it for something else, and where we can kind of say, so you mentioned Patrick's study, so we work with Patrick Shofsky and things like that, where we can say, so for something else we're doing for another sarcoma type, they're running a big trial at NCI, and we say, we've got some nice data here, would you consider enrolling these sarcoma patients in this as well? So kind of taking a bit of forethought from a right from the kind of basic biology moment of saying, what are we proposing here? So when we have our selection of targets, we think a bit about what is the reality of us getting that drug to that patient. So, absolutely the idea of repurposing is blue sky, but there is, the red tape, all those conversations, everything else. But if we think about it at the beginning, we can hopefully mitigate a little bit of that and have something we progress that stands a better chance.
Yeah, I totally agree, Zoe. And again, for some other sarcomas, working with Paul Wang and colleagues, we're trying to adopt that approach of, identifying the biological mechanisms and trying to, introduce drugs into other indications when they're actually being developed for another type of cancer. But as Zoe said, it's a tortuous route sometimes. Yeah.
Thank you. Mine sort of follows on from that, actually. I think mine's a question, but it might just be a comment. So it's a big question, really, is about how do we get the balance right? Because something that resonated with me, Zoe, was when you were saying about what you're working on is to try, one aspect of your work is trying to get the immune system to recognise the cancer drug so that it actually won't be drug dependent. Your body's actually doing that work for you. So to me, economically, as well as a somebody that's on medication, that would be absolutely an amazing thing to happen. However, I'm sure some of the funding, pharmaceutical companies obviously are in a business, make a lot of money. So I'm just wondering how we get the balance right so that there's enough money going towards your kind of research that will eventually lead us to be, hopefully, non-drug dependent for survival.
That is a really interesting question. I mean, I think, you have to bear in mind that what we're talking about here is a long way off, but also there is a necessity for the thing to do that in the first place as well, and kind of that's where some of these, you still need some of that engineering, whether it's a drug or it's the kind of engineered cell type that I was talking about. And so I wouldn't say at this moment pharmaceutical companies are panicked because we're all going to somehow activate your immune system with something that, and also it's a very kind of holistic view of what we really want ultimately. And actually the reality is, like with everything that Robin was saying, is probably we'll find a context in which something works, but perhaps it doesn't work in every context. I don't think pharmaceutical companies are worried just now, but mostly because as these things evolve, there's still a need to deliver something and to maintain something and all the rest of it. I think there's a lot of potential there for business anyway, but I think also there is a recognition from all of these companies that actually there's something they can do. And although we kind of tend to think about pharmaceutical companies as being these big monsters who just want to take everybody's money, actually, I know a lot of people who work there and they have the same goal that we do ultimately, and they are really striving, albeit with the business hat on, to actually find cures and make things better and find things with fewer side effects, and all of that comes with them wanting their drug to be the best and their drug be adopted and things. So I think actually, although it sounds like we're, two schools where we've got academia and clinical side on one side and pharmaceuticals on the other, I don't think that's actually true. And I know Robin will have constant conversations with pharmaceutical companies who want to achieve the same thing we do. So I think ultimately we have to work with pharmaceutical companies in order to deliver these things. So the CAR NKs I talked about, for example, we engineer them in the lab, but we can't produce them at the scale they need producing, and you work with a pharmaceutical company there. So we have to work with them in order to be able to deliver at scale and things like that. I don't know whether you want to add anything.
Yeah, no, I think that's a really good point. And in a way, coming back to the identification of the KIT mutation in GIST, I mean, that was an academic study. It was really conclusive, and then led to everything that's happened since then. So I think there is a collaboration there, and even I suppose the introduction of checkpoint inhibitors, it led from academic work to all the sort of industry sponsored clinical trials. And I think one of the things regarding the regulatory process, it's trying to do an international multi-center trial. It's very, very expensive these days, and the costs are just astronomical. So I don't think that's going to change anytime soon, and I think we just have to work within the system that we've got.
Yeah, you're quite right in what you say about pharmaceutical companies. When I first came into this, people would say to me, of course, pharmaceutical companies don't want to find a cure for cancer because where they're going to make their money. And I've learned it's the absolute opposite. You imagine how much money a pharmaceutical company would make if they found a cure for cancer. It would just be unbelievable. So you're right. Also what I've learned is those companies really do engage with us as patient groups as well as with the people looking into trying to find research projects that allow them to produce something that's going to help. So they're on our side. We're all on the same side, all working together, and that really is something that I've learned a lot in this time.
Just one other point regarding that. I think and again, I may be a little bit cynical, but there's a particularly regarding the experience of trying to get a drug repurposed for a very rare type of cancer. I think there needs to be more dialogue with the regulatory agencies regarding we need better ways of getting drugs approved for rare cancers, and I think this sort of, again, to use the phrase one-size-fits-all approach, that everything has to be, there has to be a randomized phase 3 trial for some incredibly rare type of sarcoma. It's just impossible, and there needs to be a change of philosophy with the regulatory agencies, sometimes I think I give them a hard time about this, but they're very rigid about the ways that we measure, evaluate response. They're very rigid. If a trial, for instance, doesn't meet the primary endpoint, but there are clear signs that a drug or a treatment works, it can be very difficult to get them to reconsider the treatment for approval. So I think there needs to be more pressure on regulatory agencies to try and change this current sort of process, which doesn't really work for a lot of rare cancers. In GIST, I suppose we're really lucky because it's one of the more common sarcoma subtypes, and a lot of these drugs work really well in GIST, so that it's quite easy to show that there's benefit. But for other sarcoma subtypes, it can be really difficult using the sort of standard response criteria to show that there is benefit. But anyway, I could talk about that all day.
I can give you some hope. Within Cancer 52, so the other charity I'm involved in, we're working on that. So could we recognise that as being an overarching thing that all cancer subtypes, are facing? So, right, next question.
Back here. My question's returning to that idea of personalised treatment. You had mentioned that to figure out a tumour profile, it could be a blood test. I'm wondering if it's just a blood test or if a biopsy is required, and if any patient could ask their MDT team for access to that, or if they need to be part of a clinical trial to get access to that tumour profiling.
Oh, forgive me, I'll go to the back, and I didn't quite hear all of that. That's why I go into the audience. And
I've got the quiet mic.
No, I'm the quiet one. It's too soft when I...
So the question was, you had mentioned about tumor profiling, and I was just wondering if that's something, is that a blood test, is that a biopsy in order to get that information, and could any patient request that from their MDT, or do you have to be part of a clinical trial to get access to that info?
So currently in the UK, for patients who are being treated with imatinib or one of the other drugs, we do the mutational testing on the tumour material, and again, coming back to Judith's point, we have that discussion with the patient regarding the mutational profile and what the best treatment is. In terms of the blood test for circulating tumour DNA, that isn't standard in the UK right now. It's, I suppose, in a way, remains to be proven whether it is effective, and that's one of the main points regarding this trial of ripretinib versus sunitinib for patients with a ctDNA profile of KIT exon 11 plus the resistant mutations of KIT exon 17 and 18. So I'd say watch this space regarding the use of blood tests, and hopefully it will become standard. But in terms of a discussion with patients, yes, of course, then we always talk about the mutational profile, and yeah. Good. Sorry, my hearing has no, my eyesight and my hearing. But
Hi there. It's handy that you're here. Hopefully you can hear me. So I'm here on behalf of my husband. He has metastatic GIST. He started imatinib 6 months ago in April. I'm going back a little bit to what you were talking about earlier in terms of side effects and of the off-patent, the fact that it's off-patent. That's really interesting to me because he's been on 3 different types since April of imatinib. And I suppose my question is, how long on each one do you know before? I don't know that we've got any grasp of whether each one has had different side effects to the other. So he's been on the last one for 5 weeks maybe. What sort of length of time should we be asking to be on one type for a few months before we get a real gauge of what the side effects might be? Because at the moment it feels a bit random all over the place.
Yeah, in a way, there's no right or wrong answer to this. And, I think sometimes there is a need for dose reduction, and that's the first thing that sort of springs to my mind in terms of managing this for your husband, whether starting at a lower dose and then seeing how things go with the lower dose and working the dose up would be a good way to...
Okay, so he's currently on 400. I think his worry would be that if you drop it, is that going to have less effect?
Well, as we discussed earlier, there's no, it's a flat dose for everybody, and everybody's different. So the way that the body metabolizes the drug will be different between different people. So and again, there's no right or wrong answer, but my take on that would be maybe to start at a lower dose and then try and gradually work
on one of the specific types, though, because that's the issue. He's had 3 different brands of imatinib in the space of 6 months. So how do you get a grip on whether one is causing more side effects than the other?
So I think they'd probably need to be on a drug for about a month to really get a good handle on that. Okay. But as I say, my take on it would be to try and adjust the dose, sometimes even adjust the time of day that somebody takes the drug, depending on the side effects that somebody experiences. So, sometimes even just taking it before going to bed or something. So there are ways.
Yeah, he started off doing that, and then he was told to maybe try at breakfast. So maybe some of the issue is that we haven't had much contact with the consultant.
Yeah, so I really have a chat with the oncologist about that.
That'd be great if we get to see him.
Yeah. So and just to try and find the best way forward. But I usually, if somebody runs into problems, try and start at a low dose and then work up.
Okay, maybe I need to speak to some other people to find out what sort of side effects they're getting and whether what he's getting is unusual or not. I don't know. Yeah,
and again, it's good to talk to your oncologist about that and about because this is all about, in a way, tailoring it to the individual person depending on the side effects and depending on the ways to try and manage those side effects. But there are definitely ways of doing that, and sometimes we even do the blood imatinib levels just to see if it's really, really high, and then clearly the dose is not the right dose for him.
So a blood test could test what the imatinib is doing, or how
high the level of imatinib is in the blood.
Okay.
But I in a way, I tend to be a little bit more pragmatic than that, and just as I say, start at a low dose and then adjust as need be, and increase when if somebody's fine on 100, then increase to 200, and just see how things go.
Right. Okay. Sorry. Thank you.
Yeah. No problem.
Thank you. Hello.
Hi, how are you?
Hi, all right, thank you.
Good.
I'm just wondering if any of the trials for second-line treatment are, you have to wait for the GIST to metastasize rather than stabilize. So if you've got stable GIST, do any of the second-line treatments cover just in case, you know, they progress, or otherwise you have to wait for the progression and then go on the second-line treatment?
So all the trials, and you know, even taking a step back, if somebody is doing well on imatinib, and the cancer's stable, then it's probably better just to continue on a treatment that's working.
All right.
So all the trials are for people where the cancer has started causing problems
After
and
they've
grown
progressed.
Again. Yeah, indeed. And as I say, if somebody is doing well on imatinib, it makes sense to just carry on with that.
Even if the shrinkage is stopped and everything.
Yeah, but if the disease is totally, if the cancer is totally stable, then it makes sense to.
Right.
And again, we've got people in clinic where the GIST has been stable for years. There's been no real change. You know, maybe a millimetre two up and down on each scan, but absolutely stable. And it's better to stay on a treatment that's controlling the disease. Yeah. I told you it was much better that people ask questions rather than me just going.
Hi, you just mentioned about imaging. Is there a preference in imaging? Because obviously you've got your CT and your MRI. They both have benefits. When you're discussing trials and recurrence, do you prefer one type? Is there one type of imaging that gives you better results? Because obviously some patients will have one, some patients will have others. Is there a point at which you say one's better than the other?
Yeah, good question. Again, I think there's no definitive answer to that. A lot of it depends on and where. Well, it's for some people, CT may be better because sometimes the disease can be difficult to identify on MRI, and the other way round. Maybe not so much for GIST, but for other sarcomas, there have been situations where, you know, tumours have been visible on one type of scan, but not on the other. One of the things with CT is the radiation exposure, and if somebody is really young, then we'd probably try and use MRI rather than CT. But if somebody is a little bit older, then the risk from radiation exposure is much less in terms of causing problems down the road. But there's no, and you know, of course, somebody may have a reaction to CT contrast, may have a reaction to MRI contrast. So again, it's very much an individualized approach. In general, I think for we'd usually use CT, usually just CT abdomen and pelvis.
Thank you.
Yeah, pleasure.
Is there any work being done to monitor the effectiveness of generic imatinib versus Gleevec?
So we are doing, we are monitoring the side effects, and in our institution doing an audit in terms of the side effects, and that will encompass, you know, if somebody has to come off generic imatinib due to the cancer getting worse. I'm not aware of any trial, you know, comparing different formulations of Gleevec, but we are looking at the impact that these generic formulations have had on patients, particularly as we discussed earlier in terms of side effects.
My question is more about the effectiveness of it, not it being tolerated, but for instance, do the generic imatinibs, are they as readily absorbed as Gleevec would be?
So I don't think there's a lot of work, looking at that prospectively in terms of the absorption of the different formulations of the drug. Again, in a way, it comes down to funding and trying to look at that prospectively. I think we'd need a lot of funding, and I think, unfortunately, in a way, for grant review, it would be a challenging. They'd say, well, this isn't exciting enough for us. We need, really cutting edge science. This is just looking at an old drug and different formulations of an old drug. So, I unfortunately, I don't think there's a lot of work looking into that.
But there is the experience of the oncology teams that are treating people.
And that's what we're looking at, the side effects primarily, but also we'd be looking at how long people are on treatment, and if there's a signal from those retrospective studies that one of the formulations is worse, then of course, we'd need to look into that a bit more. And I think that would sort of prick up the interest of external funders. Mm.
We've got time for one more question. Burning question?
I should have given an even shorter lecture in that case. So that
okay, right. Oh, there we go.
Hi. I just wanted to ask about resectioning as opposed to non-resectioning, where there's a problem in terms of like the location of the tumor. What would happen if the decision was not to have a resection for that particular tumor?
Yeah, so that's a fantastic question. I think that's really, really difficult because some anatomic locations are really, really challenging. And it's difficult to give an all-encompassing answer to that because it comes down to the individual and the individual, dare I say it, the surgeon's comfort with the risks of doing an operation. And if everything works out, then it's fantastic. But if somebody dies in the operating room, it's just awful. So I think that's really, really difficult. And you can always ask for another opinion, another surgical opinion regarding resectability. And I think, again, it comes down to, very careful discussion between the individual person and the surgeon. But you're absolutely right. It's very, very difficult. Very, very difficult. But I think getting another opinion is entirely reasonable in that context. Very, very reasonable. Great question. Yeah.